So thank you for having me. This is really a big pleasure. It's early in the morning here in Spokane on the towards the west coast of the United States. So if I am for whatever reason not clear, and I can be not clear in the morning sometimes, please feel free to interrupt my talk and ask me questions so that you can follow along. I'll be happy to take questions along the way. Um, I decided to put up this slide here with pictures of my team first because sometimes in the enthusiasm with the Q&A at the end, I might forget to thank all the people that actually do all the hard work in my lab. And so some of these pictures are a little bit older because of the pandemic. We haven't had a chance much to take new photographs. But I have a fantastic team that have really been doing all the work that I will be talking about. And I wanted to make sure that they are all acknowledged. So what I will be talking about today is research that started about almost 25 years ago with the realization that there are large individual differences in how people respond to sleep deprivation. And from that observation, from that first observation, a couple lines of research actually spun off. And it would be impossible to capture any of that in even about a 5 hour lecture, which I'm not going to try to do today. But I'm going to showcase a few examples of how that line of research has moved our thinking forward in understanding how sleep deprivation affects cognition, and how genetic polymorphisms have actually helped us along the way. So first, I need to talk about the psychomotor vigilance test, which some people consider to be the gold standard for how one measures the impact of sleep deprivation on cognition. You see a picture of the task here. There's a little on that screen there. There's a little red box, and in that red box, I know it's too tiny to see, but there is a millisecond counter there where that starts counting at 0, and then with every millisecond goes up by 1. And so within a couple hundred milliseconds, you'll see larger numbers appear there. And your job when you're doing the psychomotor vigilance test is simply to stop the counter by pressing a button. The moment you press that button, the counter stops, and it will therefore show your reaction time because that's where the counter stops. So it will be a reaction time in milliseconds that stays on the screen for about for a second, and then the little box turns black, and 2 to 10 seconds later, at a random interstimulus interval, the counter pops up again, starts counting at 0 again, and you just repeat. You press the button as soon as you see it, and thus you get in a 10-minute period, you get about somewhere between 70 and 100 or so reaction times. And if we look at what those reaction times show, and this is data from one single individual who performed the task at 12 hours awake, 24 hours later at 36 hours awake, so skipping a whole night of sleep, and another 24 hours later at 60 hours awake, so now skipping 2 nights of sleep. Then we see that at 12 hours awake, so that's normal, that's a normal day, that's basically baseline performance. We see that in a healthy young individual, which these data are from, the reaction times are very tightly distributed at around 250, maybe 300 milliseconds, very steadily so. There's a few reaction times a little bit longer here, but this is what the good performance of somebody who's paying attention for 10 minutes straight looks like, with very little variability in reaction time. So this is a healthy, young, alert brain. But that same brain 24 hours later pretty much actually shows the same phenomenon, except here and there, like for instance here, all of a sudden, out of nowhere, there is a much longer reaction time, which we typically call a lapse of attention, that had no preceding warning, and there's seemingly no after effect. It just came out of nowhere and then disappears again. There's one here, there's one here a little bit maybe, there's one here and here, but most of the reaction times are not like that. You also start to see these gaps here. These are false starts. Full starts happen when somebody's trying to do this task, trying to continue to pay attention, but is losing track of time, and presses the button just to make sure that they would respond. So we actually consider that to be a sign of effort, that they continue to put effort into the task. And so what you see is 24 hours after, later than as the first task, we now see a bit of a mixture of mostly normal reaction times with an occasional lapse and an occasional false start. And then another 24 hours later, we see this being much more exaggerated with now a bunch of reaction times that are quite long. Here's one that's more than 6 seconds long. Um, here's a few long ones, some false starts. But again, we also still see normal fast reaction times in the order of about 250 milliseconds. And so what this data tells us, and what you can see at a larger scale, if you look at data from a whole bunch of individuals, is a shift in the distribution of the reaction times, where baseline performance here in blue is very much clustered around 200 to 300 millisecond, this domain here, steady reaction times with very few longer reaction times in the tail of the distribution. But if you get people sleep deprived, you see, this is now after one night of sleep deprivation, you see that the peak of the distribution shifts only a little bit. So the mode, the mean, and the median shift only by a tiny bit, but you see an increasingly fat tail on this distribution. And now if we draw an arbitrary line at 500 milliseconds, which is what we typically do, and we just count the reaction times in that tail, which we call the lapses of attention, then we see that after sleep deprivation, we get more lapses of attention. And all of the action in what changes in this task is really in the tail of the distribution, which again is a reflection of instability in the brain due to sleep deprivation. Now, interestingly enough, people vary tremendously in how susceptible they are to this phenomenon. I'm showing you data here from 24 subjects who were, again, healthy young individuals who were kept awake for 40 hours, sorry. And so there's 24 in the total group, but I'm only showing you the 7 that are the most vulnerable or the most affected by sleep deprivation, and the 8 subjects who are the least vulnerable or the least affected by sleep deprivation. And in the group that's most vulnerable, we see a very characteristic pattern of responses to sleep deprivation across a period of 40 hours of wakefulness, which starts with virtually not much impairment at all. These are now the lapses of attention. These are the reaction times that ended up in the tail of the distribution, and higher up on this graph is worse performance. But in the first 16 to 18 hours or so, nothing much happens. But then as we get into the evening hours and beginning of the night, we see a very steep incline in performance impairment, peaking actually around early to late morning hours. Then it gets a little bit better towards the afternoon. If any of you has ever stayed up a whole night and worked into the next day, you will know that once you get past the very difficult morning hours, in the later afternoon, it actually gets a little bit easier. And that is because the circadian pacemaker, or the biological clock, produces a pressure for wakefulness that is strongest in the late afternoon. And so that actually helps you out. And so performance gets a little bit better in the afternoon, even if you've stayed awake all night, but not quite like it was at baseline. But this group here in green, the most resilient individuals, in this respect is actually much more interesting because they barely show any lapses of attention. They have a little bit of a dip in a little bit of a peak of impairment, but not much in the late morning hours, but then not much after that either. So they are very resilient to the effects of sleep deprivation. And one of the interesting questions that hasn't fully been resolved to date is what makes these people different from each other? Interestingly enough, if we ask these individuals from the same people, how do you feel about this experiment subjectively? How sleepy are you? Then the two groups who are quite different in their objective performance impairment subjectively are right on top of each other. So these two groups do not, at least from a subjective perspective, tell each other apart. Which, of course, has a very interesting implication that if you ask people subjectively, are you sleepy? They will be able to tell you whether they are sleepier than they were before, but you will not actually get a very good insight into how impaired they might be when they're asked to do a task. Now, you might think that this is only relevant to people that are studied in a laboratory like we have, and that if you go to the actual real world, that this is not much a problem. And most people that we work with in operational settings, whether those be firefighters or pilots or people that work in hospitals, doctors and nurses, or astronauts, you name it, pretty much all of these groups of people, all these professional groups will tell you that they can actually do relatively well under conditions of sleep deprivation. They do this all the time. They have learned to cope with it, and they're going to be okay. And of course, this is particularly true for people like Air Force fighter pilots, who are a highly selected group of people, very elite group of selected people, who all believe that they are invulnerable to some extent. And if we look at what happens to them, this is data from, there should be another picture here. Hold on, let me pull that up. Yeah, there we go. So from fighter pilots to fly this F-117A Nighthawk fighter plane. It's a stealth fighter plane. It's not currently used much anymore, but at the time when this experiment was done, it was the state of the art in fighter planes. And they have a simulator here that they use to actually stay current on their flight licenses when they're not actually flying missions. So in peacetime, they don't really fly a whole lot, and so then they use the simulator to stay current on their flight abilities. And this simulator was used to conduct an experiment of a simulated night shift mission that started at 11 p.m. And then they were flying at 4 a.m. and 9 a.m., 2 p.m. the next day, and 7 p.m. the next day. And they were flying a mission. And at these particular points in time, they were asked to do a number of different maneuvers. One of the maneuvers that they were asked to fly is the left 720-degree turn. So that's 2 full circles. It's very difficult to do. It's very difficult to keep your roll angle very precise doing this flight maneuver. And you can measure in a simulator, you can measure how much they deviated from the flight path. And we see this one fighter pilot here who started with really good performance, barely showed much of a deficit through this entire time being awake, through the night and into the next day. But this other pilot showed significant impairment in the late morning hours, just like we would have seen in healthy, normal individuals who are not fighter pilots, got better in the afternoon because of the biological clock, but showed a markedly different profile in this ability to fly this particular maneuver and to stay with the flight path than the one who was more resilient. And if you look at all 10 pilots that participated in this experiment and how they were performing compared to each other, we see what is actually a bit of pretty much a normal distribution, with some individuals being very resilient and some being much more vulnerable to this period of staying awake, just like we would see in normal healthy individuals who are not pilots. And so we learned from that experiment that selection mechanisms and self-selection mechanisms of people who would be expected to not be in the population anymore because they are vulnerable to sleep loss are not actually eliminating these individual differences in vulnerability to sleep loss from the population in operational settings. One of the things that we were interested in, as you can imagine, is what makes this happen? Why is this so? And because these individual differences are trait-like, and I'll show you some data on that issue later, we also started looking for genes that might predict this trait. And one gene that we stumbled upon that is actually relevant in this story is TNF-alpha. TNF-alpha is, of course, a cytokine, pro-inflammatory cytokine, but it's also a sleep regulatory substance. And it turns out that a polymorphism in the TNF-alpha gene predicts to some extent trait individual differences in how much people are affected by sleep deprivation in terms of sustained attention performance, which is the thing we measure with the psychomotor vigilance test. And so here we had an experiment where we had 88 people. Most of the people were of this particular SNP were homozygous for the G allele, but there was an A allele which is rare, that we found some of these people homozygous for that allele in this group, and there's a relatively small group of people with a mixture with one of the A allele and one of the G allele, so heterozygous. So this is the distribution of genes, and you can see that people with the A allele are, of course, much more rare than the ones with the G allele. But if we look at their performance, we see a marked difference, especially between these two groups, the GG and the AG, who are the most individuals in this experiment. And we see that people who are homozygous for the G allele, which is most of us, were more vulnerable to having lapses on average doing an experiment with sleep deprivation than those who had an allele in their genes. Here's what that looks like over time. So closer to the picture that you saw previously, where we have performance in the first this is over 36 hours of sleep deprivation now. Over the first 16 to 18 hours, not much happens, but then you get this increase in performance impairment, and then subsequently a slight improvement in the late afternoon the next day. And here's the average profile for the homozygous G allele carriers. And here's for those who carry one of the A alleles in blue. And you see that there is a difference and with the people with the A allele being more resilient to the effect of sleep deprivation here than those with the homozygous for the G allele. Now, the differences between people in this group are much wider. Here is the lowest tertile, so the lowest third of the group that is much more resilient. And here's the highest tertile, so these are the people that are most vulnerable. And the difference between the A and the G alleles is only a portion of that difference that we see overall. But nonetheless, this one gene explains 6.4% of the variance, statistically significant. And for a single gene to explain that much variance is actually rather rare. So this is one of the few genes that actually explains a significant amount of variance in this difference between people that we've been able to find so far. And I might, towards the end, might get back to you a little bit more on why this might be so. Well, before I get to that part, though, before I get more into the underlying mechanisms, there was another observation that Lily Skyke, a graduate student of mine, made a couple of years ago, where she had a larger group of now 168 people that had were genotyped for this particular gene. And again, we have most of the people are carriers of the homozygous for the G allele. We have some carriers of the A allele. There's only one that is homozygous for that. And again, we see differences between these two groups in the vulnerability to sleep deprivation. Again, the G allele carriers are more vulnerable to sleep deprivation than the A allele carriers. But as you might have noticed in the earlier graph, there's a small difference also even at baseline already. So even at baseline, when there's no sleep deprivation, carriers of the A allele are just ever so slightly better yet at performing this task than those who have the homozygous for the G allele. So there's something about these people that's not just related to sleep deprivation, they're just overall better at sustaining attention on this task in general. And then she looked at what happens with these people's sleep. And she looked at what happens in these people's sleep both at baseline sleep and also at recovery sleep, and found that regardless of whether you looked at baseline sleep or at recovery sleep after sleep deprivation, those carriers of the A allele had less delta power and less theta power in the EEG of non-REM sleep. And what that means is that, well, that's typically interpreted, I should say, is that the A allele carriers had less pressure, homeostatic pressure for sleep in their system all along. So they were just actually as if it were that they were more well rested than the G allele carriers, despite the fact that the amount of sleep that they got was actually the same. So there is something appears to be about these A allele carriers that makes them either more efficient at recuperating while they're sleeping, or they get more recuperative value out of the time that they spend in non-REM or slow wave sleep, or something else related to that makes them more efficient in their recuperative power of sleep, that actually gives them an advantage that is not just related to sleep deprivation, but is an advantage overall. And we now believe, for other reasons that I won't have time to talk about, that this is an effect of an allostatic process, which in the G alleles carriers or the G allele homozygous people actually makes them more vulnerable to sleep loss over the long run, because they are not getting enough recuperative value out of their sleep, and then they become more sensitive to subsequent sleep loss as a long-term enduring feature. And unfortunately, again, the G allele is the most common, so that applies to most of us. We all carry, apparently, we all carry, or most of us carry some sort of a underlying sleep debt with us a little bit, even if we get enough sleep, because of the genes that we carry. This is a story for another day, but it's an enticing piece of information that, of course, wakefulness and sleep are tied to each other. And you can't really talk about the effects of sleep deprivation without also looking at the sleep associated with it. But I'm going to leave that part of the story behind and focus a little bit more on what happens during wakefulness. Um, Colleagues of ours at the Federal Aviation Administration did a transcriptome analysis on people that had been sleep deprived in our lab, for whom we took blood samples. This was now in the last 24 hours of a 62-hour sleep deprivation experiment. So this was after having already been kept awake for a whole night and then staying awake for a second night. Looked at the blood samples from that experiment and did a transcriptomics analysis, so looked at the gene expression profile and found that there were 212 genes in these people's transcriptome. They were significantly altered by staying awake. So there was 212 genes that showed changes because of being awake in the transcriptome. But out of those 212, there were 28 genes specifically related, not so much to time awake alone, but specifically related to performance on the psychomotor vigilance test. So the ability to sustain attention was tied to the expression of 28 different genes specifically. And here is a network plot of those genes. I'm not going to try to explain what each of the genes are and how they hang together. And keep in mind that this was a pilot study in this regard. This is just it was a fairly large sample. I forget how many, but it's not quite enough to really know what's going on here. You need much larger samples for that. But it's nonetheless enticing and interesting to think that there are some genes specifically related not so much to just being awake, but also to what you do while you're awake and determine the outcome. And that turns out to be a very important observation for other reasons as well. As I mentioned previously, I was going to talk a little bit about more about this trait aspect of this vulnerability to sleep loss, and I'm going to do that right now. What we did about 20 years ago, or a little bit more than that, we started an experiment where people came to the laboratory multiple times. They actually came to the lab 4 times, one for an adaptation session, one time and then 3 times to be sleep deprived. And out of those 3 times when they were sleep deprived exactly the same way under the exact same circumstances twice. There was a 3rd time then they were sleep deprived, and they were also then already pre-sleep deprived, pre-sleep restricted before they came to the lab. So they were already tired before they even came to the lab. I'm not going to show you the data of that part of the experiment today. It's not particularly important right now. What I'm going to show you is the data from the 2 times when people came to the lab that were exactly identical. They were being kept awake for 36 hours. They performed the psychomotor vigilance test, but this was a 20-minute version of the task every 2 hours while they were in the lab. And you see here the data, the averages of the last 24 hours of that sleep deprivation period for the first and the second time that they were in the lab in the dark and the light blue boxes, right? So every subject who's on the horizontal axis here has 2 points. One is the average of their number of lapses on the psychomotor vigilance test the first time they came to the lab, and that's in dark blue, and one is the second time, which is in light blue. And what you see is that pretty much every individual, with only one exception, had very similar response to this experiment the first and the second time. The data points are always very close together. But there's this massive spread between those people who were most resilient and had very few lapses on the left, and those who were most vulnerable, who had many lapses on the right. There's one subject who was not very similar to themselves, but everybody else is very close and very repeatable experiment, basically. They showed pretty much the exact same thing the first and the second time they were in the lab. And there's at least 2 weeks in between these experiments. So that is one part of what told us that there's a trait here. This is a systematic individual difference. It's repeatable, and it turns out it's also robust to changes in circumstances. Not going to show you that today. And it repeats itself no matter what you look at. If we look at digit symbol substitution task, which is a completely different kind of task, where you have to match numbers and symbols and be as fast as possible and do as many of these as you can in a 3 or 4 minute time span, 3.5 minute time span, we see the exact same phenomenon. So wide differences between people, but the average performance impairment during the last 24 hours of sleep deprivation is very similar from the first to the second time within each individual, right? So again, there's a trait-like phenomenon here. But what might also catch your attention is if you look at the rank order of these individuals, the same letter U is the same person here and here, that this person is highly resilient to sleep deprivation while doing the psychomotor vigilance test or the PVT, but is actually among the more vulnerable participants when doing the digit symbol substitution task or the DSST. And so, even though this is a trait-like phenomenon, it turns out that where you end up on the resilience to vulnerability spectrum depends not only whether kind of what kind of person you are, but also what you're doing, right? And so if you look at all the letters here, there's actually a correlation in rank order between the same people here and here that is very close to zero. And this diagram here, your vulnerability to sleep deprivation in this diagram here does not at all predict where you end up in this graph here. And that, of course, caught our attention because if we're looking for genes that are explaining, that may be explaining vulnerability to sleep loss, but it also depends on what you're doing, then that complicates the search for genes quite dramatically. Now, to continue, I want to actually take you in a slightly different direction here and walk you through a particularly interesting accident that happened now almost exactly 30 years ago um, in a civilian plane that was resupplying the military base, American military base on Guantanamo Bay at a time before this naval air station became infamous for other reasons. Um, this particular accident is quite well known in the world of accident investigations, uh, and I'll tell you in a minute of why that is so. But what you need to know about this accident is that so if you fly into Guantanamo Bay Naval Air Station, the airfield is right here, that horizontal stripe, red stripe that you see there. And to approach that airfield, there's two ways you can go. You can actually come from what would be on your screen from the right and fly into it along the coast, drop down into the airfield and just land. That's an easy landing to make. But to get to this point, you actually need to come around and have a much longer flight route to make this approach. So it's the easier one, but it's also the longer one. It's more difficult. It's more time consuming. There's a faster way to go, and you can just fly in from the south and go straight to the coast, get to this point approximately here, and then make a very hard right, drop down, and get onto this airfield. But to do that, you actually have to then watch for a strobe, which is about here somewhere, to know where to make that turn. Because if you don't, and you fly too long, and you keep going, then you get into Cuban airspace, and that would create an international incident. So this is a landing that's faster. You go straight up and make a hard right and drop down. But you need to watch for this strobe here and not get that wrong, because otherwise you get into the wrong airspace. And this is a more difficult landing to make. Now, the air crew that was resupplying this base, that was flying in that day when this accident happened, had been on a long day already. This was, I think, their the third in a long range of flights that they were doing. And they were getting towards the end of the day. They were tired. They had initially been told because of weather they needed to make the roundabout and the longer approach, but the weather cleared and they got permission to do the faster approach. They had a little bit of discussion and decided that they really wanted to go to bed as quickly as possible. So they decided to do the more difficult approach to get back there sooner, to get home soon, to get to their sleeping quarters sooner. And so that's what they decided to do. What they did not know, that nobody knew, was the strobe that was supposed to tell them to take this hard right was not actually on that day. It wasn't actually working. Nobody knew that. What I'm going to do now is I'm going to play to you the transcript of what happened in the cockpit upon this approach. So the captain is looking for the strobe. He won't find it. He says, where's the strobe? And the engineer says, right over there. But the engineer is wrong. It wasn't actually there. He was seeing something else. Captain says, where? First officer, who also thinks he sees it, says, right inside there, right inside there. Then the engineer says, we're losing our airspeed. We need to get our airspeed back on. Captain says, where's the strobe? First officer says, right down there, but it's not true. Captain says, I still don't see it. The engineer is starting to curse and says, we're not going to make this. The captain just keeps going. Where's the strobe light? The first officer says, right over there. Captain, where's the strobe? Where's the strobe? First officer, do you think we're going to make this? Captain, yeah, if I can catch the strobe light. Officer is now saying there's problems. Engineer is starting to shout. Now there's a stall warning. Captain keeps like, I got this. Stall warning, stall warning. Captain says, I got this. Back off. Maximum power. Nobody knows who said that. It was intangible. And then there it goes, and it crashes. And so they're screaming on the cockpit recorder, and what ended up happening is this plane crashed. And it is notable in the area of accident investigation because this is the first time where the National Transportation Safety Board in the United States, which is the organ that the agency that investigates these accidents, officially proclaimed that fatigue or sleep deprivation was a cause of this accident. They don't normally point that out because unlike things that are broken and are tangible causes of accidents, fatigue is something you just have to estimate. You can't find it in a pile of rubble. But they were so certain that this was a fatigue-related accident that this is the first one where they officially made that a determination, that they said the impaired judgment, decision-making, and flying abilities of the captain and the flight crew due to the effects of fatigue were the root cause of this accident. And so what we learned from this accident, from other things that have happened since then, is that sleep loss does not only cause this inability to sustain attention, but it also causes a loss of cognitive flexibility, and it makes people perseverative. They stay stuck on a solution that is no longer effective. As we saw with the pilot just keeps saying, where's the strobe? I need to find the strobe. Even though the first officer and the engineer that were on board were saying, no, we need to pay attention to our airspeed now. When you go to pilot school, you learn that the first thing you need to do is aviate. Second thing you need to do is communicate. And the third thing you need to do is navigate in that order. But the captain got stuck on navigating and completely forgot to actually fly the plane or to communicate about that. And so the captain became perseverative, and there are other instances that we know about where similar problems have caused major disasters. This, of course, Chernobyl was a similar phenomenon where people got stuck on something that wasn't working, that they were not recognizing circumstances had changed, and kept doing what they were doing as if there was no problem. And it can lead to major catastrophes and also to more mundane accidents that happen every day. And it's unrelated, appears to be unrelated to the sustained attention deficit that we talked about previously. So we decided to look into this issue more deeply because maybe that is one reason why people can be vulnerable to sleep deprivation in one particular domain, but can be resilient to sleep deprivation in another domain and vice versa. And I'm going to walk you through a laboratory task that captures the same phenomenon, but in a different way. So in this task, you are shown, or you have 4 two-digit numbers, so 42, 63, 81, and 15, for instance. And if these numbers appear on the screen, you have to press a button. We call those the go set or the go response set. There's another 4 two-digit numbers that are no-go stimuli. If you see any of those, so that might be 27, 30, 34, 41, and 92 or something like that. If you see any of those, then you're not supposed to press a button. So those are no-go stimuli. And the other thing about this task that's important is we don't tell you in advance which ones are the go and which ones are the no-go stimuli. So you might see the number 42, and the first time you see it, you have no idea whether this is a go or no-go stimulus. So you might decide to press the button just in case. and the computer will tell you that this was not right, that this is a no-go stimulus, and please don't do that again, right? And so once you've seen that, you can now learn that 42 is a no-go stimulus. And the next time you see it, if you remember that, you will then not press the button. So people learn as they do this task, which of the stimuli are the go and which of the stimuli are the no-go stimuli. But then unbeknownst to them, after 56 or 60 or 64 trials, depending on which version of the task we are administering, we actually flip the contingencies. So now, even though 42 was previously a no-go stimulus, for instance, it's now become a go stimulus. And so now, if you see 42 and you don't press the button, the computer will tell you that you were wrong. And you're supposed to learn from the feedback that the computer gives you that something has changed, right? And so now there's something has changed and you need to change your responses. You need to adapt your understanding of this task. And so we measure how people do that for another 40 trials. And that's how this task works. And so we call this a reversal learning paradigm. And the reversal learning paradigm helps us understand what goes wrong in terms of perseveration and when you're sleep deprived. So here's data from this task. Now, I'm going to be a little quick about this. This is the second time actually that they see this task. So they've done it once before. They've done it once before at baseline. But we have two groups of people now who are doing it first at baseline, but then half the sample was sleep deprived, and the other half of the sample was actually well rested. And here's what we're looking at right now is their performance on this task during the second time they see the task when the blue group was well rested and the red group is now sleep deprived. And so you see that in these at cross trial blocks, this is before the reversal, you see an improvement in the number of hits, so they get more of these stimuli right, and the fewer false alarms, so fewer that they do wrong. Whereas the group that's sleep deprived is already struggling a little bit. They're not learning as fast. They're not improving as fast, not making as many hits, and not avoiding as many false alarms beforehand. Then we get the reversal, and then what we see after the reversal is that the well-rested group, they of course are thrown off by the reversal because that's the whole point. But they learned quickly to understand that something has changed, and they pick up from where they left off and get better at the task again. More hits, fewer false alarms. But the sleep deprived group is completely thrown off and never recovers. This is chance performance. They have no idea what's going on anymore. They're completely stuck on this task. That's in part because they never really learned what was going on to begin with very well, but the reversal throws them off completely. They cannot do this task. The effects are so detrimental that we had never seen any effect sizes this big before. Now you might ask yourself, is it so difficult to do this task when you're sleep deprived because you're missing information, because you have lapses of attention, you're not sustaining your attention, and so it is a lapses of attention phenomenon, just like on the psychomotor vigilance test, perhaps. And we can simulate that by having well-rested individuals do the task, but actually hiding some of the feedback from them. We might just give them either all the feedback or only 80% of feedback or only 60% of the feedback on the task. And if we don't give them the feedback, that would simulate what would happen if you just missed the information, if you were just lapsing. It turns out that performance on this task, when you're well rested, but you are masked on this feedback with only 60% of the feedback coming through, you still do better than people who are sleep deprived. So it's not just that you're losing information, something else is going on. And in fact, if we ask people if maybe they just forgot what the stimulus was, so that when the computer tells you that you're wrong, that 42 was not a go stimulus, but you don't remember that 42 was the number you were responding to, then that would also be a problem. But it turns out that people are almost perfect in remembering what the stimuli were that they saw. When we ask them, they know exactly what they saw, at least more than 95% of the time. So it's also not a loss of memory that's happening, at least not in that sense. It's something else. And what that might be, we learned from looking at these data a little bit more carefully and also looking at skin conductors responses. What I'm showing you here right now is performance on this task as an overall discriminability score. The first, the second, and the third time they saw the task. This is the well-rested group. This is before the reversal. This is the well-rested group here after the reversal. And you see that every time they do the task, they get better at it. They learn the contingencies more rapidly. Here's the sleep-deprived group, and you see that before the reversal, but especially after the reversal, their performance is really poor when they're sleep-deprived, just as I already showed you previously. But if you look at skin conductance response, which measures the salience of the feedback, so the importance that the feedback has to people that are doing this task, we see that when people are sleep-deprived, the salience is dampened. As if the information about the feedback that they get, about being right or wrong about these stimuli, is less important to them. They don't really seem to, it doesn't get the same level of top-down attention from their brains as if it really matters when they're sleep deprived as it would otherwise. So that was the first hint that we got that something else is going on. This is an attention problem for sure, but it's more like top-down attention. It's more like being able to focus on the information that matters. Not very different actually from what we saw with the pilot, right? With the captain who just kept focused on the strobe and forgot to realize that there was much more important information to be concerned with. So turns out that this phenomenon is also genetically determined to some extent, but it's a dopaminergic phenomenon. There's a gene, an enzyme called COMT that removes dopamine from the prefrontal cortex. It has a pretty common single nucleotide polymorphism. Val to mat substitution of this gene is quite common in the population. And we divided our group of 41 individuals into the different genotypes on this particular gene. You see that here. Most of them are vel-mat heterozygotes, but the homozygous met-mat and vel-vel are about the same representation here. And at baseline performance on this task, the first time they see this task, there isn't really much of a difference. This is not statistically significant. When they do the task again, as you have seen before, they get better at it. But if this is the well-rested group here, there is again no significant difference. There's some variability, but this is not statistically significant. But when we look at performance on this task in a group that's now been sleep deprived for 38 hours, for 30 hours, sorry, then we see that there's a group of met-met carriers that is almost just the same as if they were well rested. But the homozygous vel-vels are almost completely blown away. And again, this is the group that their performance is even worse than by chance. And so there's something dopaminergic about how you respond to sleep deprivation when you're doing this task. So it's genetic underpinnings, and it actually pointed us in the direction of the dopaminergic regulatory system, which is a top-down regulatory system or an arousal system, if you will, being an important player in this story. If you look at what happens to the psychomotor vigilance test with this same gene, you don't see much of a difference at baseline or well rested performance, but at sleep deprived, you do see differences, but it's actually a different genotype. It's the vel-mat heterozygous ones that are the most vulnerable in terms of sustained attention, which is a very interesting dissociation between the psychomotor vigilance test and this reversal learning go-no-go reversal learning task. So to summarize, and I'm skipping a couple steps here because I'm going to be running out of time, but there are bottom-up effects on information processing, which is captured by the psychomotor vigilance test, and top-down effects on attentional control, which are represented by this reversal learning task, that are separable and must have distinct underlying mechanisms. And I'm going to, in my last couple of slides, I'm going to walk you through some further new insights that we only gained just a few years ago on what might be going on with that reversal learning paradigm. And this has to do now with an aspect of memory that is relevant in the reversal learning task and not so much in the psychomotor vigilance task. In this what we call item and source memory task, we presented people with words that were spoken to them by either a female or a male speaker. So the word table spoken by a female speaker was presented, satisfied by a female speaker, poison by a male speaker, and there were a bunch, 60 words altogether that were presented in sequentially to the participants in the study in the study phase of this particular experiment, this particular task. And then subsequently in a test phase, they were now presented with 120 words, 60 of which were the words that they had seen before or heard before, and 60 of those that were new words. These were now not spoken, but they were shown in writing. And so the word table might be presented. And if you remember correctly that you saw this word or you heard this word before, you would say yes. And then you would be asked, was this word spoken by a male or a female speaker? And you might say male, which in this case would be wrong because it would have been the female speaker that actually said that word. And then you are going to be asked how confident you were about remembering the source or the context in which you saw this word, right? And so this task is all about remembering not just items, but also remembering the context of those items as they were presented. So it's about remembering the source. And what we see is we look at here is well rested. Here's the first time you see this task during baseline, and the second time you see this task, which is now either well rested in the control group or in the sleep deprivation group. We see a significant effect of sleep deprivation. On item memory, people do much worse in this task when they're sleep deprived than when they're well rested. But if you then look at the source memory for those items that they were correctly remembered, so only the items that they actually remembered, and you ask, were these the male or the female speaker? So what was the context? What was the source? You also see a deficit on top of the deficit of the item memory. The source memory is separately also affected. And so you remember less of the context of the words that you had seen before. And so the source memory is separately affected. And to show you that is really separately and not just another version of the item memory problem, we look at serotonin transporter gene. And the serotonin is particularly relevant in this story because that is a neurotransmitter that's heavily represented in the hippocampus, where binding of memories actually occurs, where items and source memories are coupled together. And we see that for source memory, there is a significant effect of genotype that carriers of the long version of this particular length polymorphism of this particular gene, they do much more poorly on the source memory than those of the short version, who actually do just as well as if they were almost well rested. And so this, again, this is very new data. That this is only 2 weeks old. Tells us that there is another regulatory mechanism here now related to the hippocampus most likely, and serotonergic in nature, that gives us yet another dimension of a phenomenon underlying these individual differences in vulnerability to sleep loss. And in this particular case, related to the binding of information from items to sources. And we think that is actually relevant in the reversal learning paradigm, because in the reversal learning paradigm, you are supposed to bind the number 42 or whichever number you were talking about to a go or no go response. And so this binding phenomenon, the item related to the context in which it was presented, is actually a critical underlying phenomenon. And we believe that perseverative errors and loss of cognitive flexibility, which is ultimately what that means, is at least partially related to this effect of sleep deprivation on binding in memory. So long story short, and I know I'm running out of time here, we think now that there are two predominantly separable, possibly more mechanisms of how you are vulnerable to sleep deprivation. We think there's this neuronal mechanism here in this zoomed in box of neurons communicating with each other. Whenever there is information being processed, there is release of ATP into the extracellular space. Long story short, that binds to the nearby astrocytes and releases a number of sleep regulatory substances, of which TNF alpha which we've talked about previously, is one of them. And in this cascade of molecular events in the extracellular space ultimately causes an adenosynergic uh, minimization of the ability of this neuron to process information, basically puts the neuronal assembly that these neurons are part of into a sleep-like state, if you will, and the information processing temporarily stops, and that generates a lapse of attention. Long story, very short. But there's also these arousal systems that are top-down, controlling our ability to exert attention and bias our attention, that ultimately determine how well we can guide our attention to things that really matter, and potentially also um, uh, affect the hippocampal ability to bind information. And those things happen in parallel. And so we've got these two competing and parallel mechanisms by which sleep deprivation can impair performance, bottom up and top down processes. These are both relevant, and they, at least in part, would explain why it is that people can be vulnerable to sleep deprivation in one cognitive domain and resilient to sleep deprivation in another cognitive domain. And they can both be traits, and they can coexist in the same people at the same time. So with that, I realize, shared a ton of information with you. I'm going to end it here. These are the funding agencies that have kept our lab open. I'm especially grateful for the ones that continue to fund us through the pandemic, which we're still feeling the effects of, as you might imagine. We couldn't have done any of this without the support of these agencies. And with that, I end it here, and I'd like to thank you all for your attention.